Welcome, everybody. How's everybody doing today? Good, I hope you're enjoying your day. Enjoying the lovely cool weather outside. I know I am. Can you hear it? No? Yeah? Okay. I know I'm enjoying this weather. I told you all last time, for those of you that were here, that I'm from Buffalo, and I'm so happy that I could just get to admire the snow from afar on the mountains. It's spectacular. So welcome tonight to the Saddlebrook Science Cafe, sponsored by the University of Arizona College of Science and the Desert View Performing Arts Center. Our series, Human Disease Pathways to Cures, in collaboration with the Department of Molecular and Cellular Biology at the University of Arizona, has a great series lined up this fall and a few more events in the spring. Thank you to the Desert View staff and all of you in attendance for being here tonight. I'm really excited for this talk. What we're going to do tonight is similar to what we've done in the past, so 45-minute talk with an audience Q&A. So feel free at the end to raise your hand for questions. I'll come around with the microphone to those of you that might have a question. And there's also evaluations in the back on the table. If you guys have a minute, please feel free to grab a pen and fill one of those out at the end. We really, really would appreciate your feedback. There are also a lot of materials on the back table for our different outreach facilities, and then also about Science City and the Tucson Festival of Books, so feel free to take those home with you. And also, just a reminder, we have a lot of other science cafes going on this week and next week and the following week throughout Tucson at the Tucson Botanical Gardens, Magpie's Gourmet Pizza, Borderlands Brewing, and then at Tumamock Hill. And if you can't make it to those in person, we stream them online on our Facebook page if you just look up the University of Arizona Science. Tonight's speaker, Dr. Carol Dieckmann, is a professor in the Molecular and Cellular Biology Department at the University of Arizona. Dr. Dieckmann graduated with a Bachelor's of Science in Biological Science from the University of California, Irvine, and a PhD in Biology from UC San Diego. She also did a postdoc fellowship at Columbia University. Dr. Dieckmann's lab focuses on understanding the coordination of organelle/host cell interactions. Two different systems under study are one, the intersection of fatty acid and lipoic acid and glycine metabolism in mitochondria. Children born with defects in fatty acid and lipoic acid synthesis and glycine breakdown are very sick and most often die at young ages. The synthetic pathway crosses the breakdown of the pathways. They're trying to understand how these are regulated under different nutritional conditions. And two, eyespot assembly and placement in the single-celled motile green algae, Chlamydomona. Sorry. Clammy can breaststroke towards light to optimize photosynthesis. Her quest is to figure out how a newly divided algal cell constructs a new eye with the help of a chloroplast and precisely place it in the asymmetric position needed for function. Some fun facts about Dr. Dieckmann. She's been at the University of Arizona for 35 years. She has a 9-year-old Springer Spaniel named Delia, and she tap dances, and she grew up near the beach in San Diego and loves to swim in the ocean. So thank you all for being here. Please help me welcome Professor Carol Dieckmann.
Hi, everyone. I'm glad there's no snow, but I am going to have a picture of snow just for all of you who are far away from snow and miss it so much. All right. So I think there's some sort of false billing for my talk. You know, I'm in this series supposed to be about human health, and I'll try to tell you a little bit about how this project interacts with human health very distantly, but it's much more fun to talk about than the other project, which really does intersect with human health. Looking at pictures of really sick children and trying to explain biosynthetic pathways with arrows just wouldn't be very fun. But this topic is really fun, and I hope you enjoy it tonight. Okay, so the title of my talk is Seeing the Light. These single cells that you see sort of flying across the page are Chlamydomonas cells, and they're lit up with fluorescent light. And you can see 3 different colors that light up different proteins, and as we go through the talk, you'll learn what they are. But you can see, even in this photo, the little red dots, those are the eyes of the algae. They actually see light, and they direct the cell to swim with the appendages that you see there called flagella. So they're really cool cells, and I hope by the end of the 45 minutes that you'll think that's true also. Okay, so you're asking yourself, what did I get myself into? She's not going to tell us about cancer. She's not going to tell us about ALS. She's going to tell us about pond scum. So why should anyone study pond scum? And why should you study the eye spot, as we call it, in a single swimming cell that comes from any old pond? And the reason is that I already told you, I think they're cool. I'm from San Diego. We used the word cool when I was growing up. So I think there's probably some of you that used cool also, or still do. I think they're cool because they do things that are really interesting, and we'd really like to understand how they work. And what are those cool things? They swim, which is pretty cool. Single cell, they can swim. They have those appendages that you saw flying out from the sides that are called flagella. And almost every cell that we have in our body has some kind of cilia on it, which is the same thing as a flagellum, or 2 flagella, or more flagella. And they wave around, most of them. Some of them don't wave at all. And they have different functions, and they're very important for many processes in all of our cells. They also see. These eyespots have molecules in them that absorb light and direct their behavior of the cell, the swimming behavior of the cell. These light-sensing molecules are called rhodopsins, and they're very similar to the rhodopsins that we have in our retina and that detect light in our own eyes. So they're interesting for that reason. You saw that the cells are green. Pond scum. I'm going to show you some plates. There's some sitting out in the lobby. Really bright green, and they are plants, and they do photosynthesis. So many people use Chlamydomonas as an experimental organism to study a very basic process of photosynthesis. It's not something that I do. There's really 3 classes of cells in the world: the archaebacteria, the bacteria, and eukaryotic cells. All of our cells are eukaryotic cells, and all eukaryotic cells have compartments. So what could we make an analogy to? Like a really big purse, really big purse. So if the purse was like a cell, and all the little pockets in your giant purse had a very specific function. I don't know, I'm older now. I have to put things in exactly the same place or I can't find them. Maybe you have some schemes too. You always put your keys in the same little zippered pocket in your purse. Well, cells have really special places that they put things in the cell. And this is one of my passions. It's really the thing that ties both of my projects together, is to understand very basic things about the compartments inside of cells. We use this alga because it has a very simple genetic system. We don't have to breed people to figure out what's going on. We can just have Chlamydomonas do sex. It's really easy. They're not called males and females. They're called plus and minus, but they do fuse and make zygotes and then start dividing again. Many of the people that are speaking in this series, I think Dr. Zarnesky was here last month, and she gave a talk about fruit flies. Fruit flies are what we call a model organism. She studies ALS. It's really cool that she can study this terrible disease in a very amenable genetic system, a fruit fly system. And this is why I use Chlamydomonas. It's a very good model system for what I want to know. Okay, so why do I study swimming, seeing green algal cells? Because they have compartments. This is a cartoon of a single Chlamydomonas cell. And all these different labels label different compartments inside the cell. And the quest that we have is to understand one particular compartment, the eyespot. Here it's cartooned as this 2 layers of these carotenoid-filled particles. Carotenoids, what are those? Carotenoids are the orange pigments that make carrots orange. And they also make the eyespot orange. So we have that in common with carrots. They have that in common with carrots. So the question is, how do they make this complicated structure? And it is very complicated. And how do they put it in a very specific place in the cell? Now we'll see how much time I have. I've decided these are really 2 kind of different stories about how you build an eyespot and how you place it. And when I started out, I was most interested in how they got built, because some of the components of the eyespot are found in the chloroplast, which is where this big cup-shaped thing that is responsible for photosynthesis. And so these pigment granules that have the carrot color are inside the chloroplast. And then the photoreceptors, these molecules I told you, the rhodopsins that actually absorb light, are in this outer layer of the cell called the plasma membrane. And so the compartment, the chloroplast, has to cooperate with the cell to make a brand new eye. Well, that's the story I'm not going to tell you today. But one thing you can see from this picture is if we cut the cell in half from top to bottom, and we folded this half over on top of the other half, pretty much everything is the same. Some of the things are random, these little red things, which are mitochondria, which is what my other project is about. And they're sort of randomly placed, but everything else is sort of symmetric around this axis. The exception is the eyespot. There's only one, and it's only on one side. It's like, what? How does that work? So that's the story I'm going to tell you about tonight. Why is it important to know where things are in a cell? Why is it important to the cell? Because if the cell compartments aren't built correctly, they don't work. That's sort of obvious. Maybe it's not so obvious that if they're in the wrong place, they most often don't work. Okay. And I've given 3 examples on this slide that are from the human body. One is your gut cells. So these are the cells that line your intestines, your small intestines. And you can see that this side of the cell, and this is the side that faces out into where your food goes through, has all these little fingers. And it increases the surface area of the gut cell and gives you more surface area to absorb the food that you've eaten. Right? The other opposite this side, that where it's in contact with the food that's coming through your intestine, is where the nutrients pass through the cell. They go through this basement membrane and go into your bloodstream. So this is how your nutrients get from your gut into your bloodstream and go all around your body. Well, if this got goofed up, you'd be in bad shape. Okay, rod cell. What's a rod cell? A rod cell is a cell that absorbs light that's in the back of your eye and your retina. So this is a cell from your eye. And it's the very important part, are all these little stacked up layers in this part of the rod cell. And this is where the rhodopsins are that absorb light. So if they're not out here and stacked up, you don't absorb light and you can't see. So it's a very important thing. You can't just have your rod discs distributed all through the cell. They have to be stacked up in this way. And the rhodopsins are all made in this part of the cell, and they get transported through this funny little narrow place here out and stacked up. So the cells have to be organized properly. And then the last one, I think you heard probably a lot about nerve cells from Daniela. But nerve cells are extremely organized. There's different parts. This is the cell body where all the metabolism goes on. And these little projections here are the dendrites, which pick up signals from other neurons. And then the signals go down the axon, and there's these little projections here, which project onto another neuron. So it's really important that these different places in the cell, it's like having an address inside the cell, okay? And all of these things have to go to the right place. Okay, so a little bit about snow. Here we go. But we'll start with ponds. So this alga that I've shown you a few pictures of is called Chlamydomonas. It's ubiquitous across the entire world. It's found in many different places. The strains that we use in the laboratory were first isolated from muddy fields in Minnesota in the 1940s. So they grow in soil, mucky soil. This is a pond in someone's backyard in Mexico. And so you know, we're talking about pond scum. It really is pond scum. This is Chlamydomonas. How many of you are from snowy places? Yeah. So have you ever seen the kind of pink snow, or sometimes they call it watermelon snow? Snow that hasn't been trodden on multiple times around the house or whatever, but out somewhere where it's a little less disturbed, and you'll often find this pink snow. So the organism here is Chlamydomonas. It's a strain of Chlamydomonas. It does photosynthesis just like the green ones, but it has a lot of red pigment, so it makes the snow look pink. So if you're homesick for snow, there you go. That's all you're going to get for tonight. Oops. What do I do? We tested these. What do you think, Colin? Yeah. Yeah, I did test it here. Should I start my... I could start it over again. No, it's the next slide. This is weird. It just usually takes a few seconds and it comes up.
That's really bizarre. It worked earlier. Yeah.
We ran all of the videos.
Yeah, well, maybe we should
Start it over.
out. Yeah.
Okay. Let's escape. Should I just start the show over again, or do you want me to back up?
That's really bizarre. Maybe we can. Sorry, folks. That's really strange. This video. Let's see if the next ones work.
I already tried one of them and I don't think it'll work. So,
Sorry, folks.
well, this is really sad, but what you're going to see is what we see when we put a drop of pond water on a slide underneath the microscope and use magnifications is about 300-fold. And what you were supposed to see is little green cells that are just zipping all over. And there are probably about 40 cells in the field of view at any one time. And they really swim fast. So they would go across this slide in maybe a couple seconds while you were watching it. How fast do they swim? They swim fast. They beat their flagella about 60 times a second. All right, so they go fast, and they go zipping across just a couple of seconds if you're looking under the microscope. All right, I'm sorry about this. Okay, we're going to try again. Oh, do you think I should go out? Oh, okay.
Yeah. Because when we reopened your computer,
Ah,
it
excellent.
didn't connect back to the internet.
Okay, so let's just talk about this slide while we're looking for our connection. This is actually a photograph I took myself with a microscope, and it's one Chlamydomonas cell. And I'm asking you, how do they swim? I think I've already alluded to that. So they have these appendages that look like feelers or antennae, but they're not antennae. They're actually, they actually are the motor for the cell. And so what do you know about, do any of you know about cells, flagella, and how do they work? How do they work? You can yell out the answer. Sort of like sperms. So if they wiggled, which direction would the cell go? It would go down, right? And most flagellated cells, the flagella wiggle behind the cell and push it. In this case, it would go down. You just wait. We're going to go back, I think, and see if we can watch the video. Yeah. Whee! Yay. What an easy solution. Of course, it was the internet. There you go. Okay. So now you know what they look like? All right. And that was real time. They weren't speeded up. They weren't slowed down. That's exactly what we would see. All right, so I asked you this question, and you answered, and you said it's going to swim down, and most flagellated cells would do exactly that. But Chlamydomonas does the breaststroke. Yeah, that's complicated, right? It's not just wiggling. You got to get both of them to do the thing together. Watch this. Now, one of the things I want to point out is this is one cell. It doesn't have legs. This is what's called a micropipette, and the cell is being held in place so you can make the video by the micropipette. It's like a suction micropipette. And then they were able to take the video, and this is just an inverted color on the right-hand side. So watch this. It's awesome. I love this thing. I could watch it all day. Isn't that great? It's so great. And I want you to see something. Can you see that maybe the left one is a little bit different than the right? I'm going to show it again. It's very quick. A little bit different, right? A little bit different. So you'll see in a minute why that's important. Okay, so the question is swimming random? Do they just do this crazy swimming all over the pond just for fun? Maybe. Maybe it's fun to swim. I think it's fun to swim. But, watch this. If we put a light, if we have Chlamydomonas, this is a petri dish. And this is in liquid. It's just like, basically like pond water. And they're in the dark. They'll be evenly distributed as they are here. And then we turn on a light on one side of the dish. This is what happens. Wow. So this is a 3-second video. It's been speeded up 240 times. So I told you that they swim really fast, but in these petri dishes, they're about 3.5 inches apart in diameter. So they have to swim really far when you're only 5 micrometers in diameter. Okay, so these guys are really small. We can't see them without a microscope, right? Sort of like the head of a pin. Okay, so this behavior of swimming towards the light is called phototaxis. And that's exactly what it means. It means swimming toward light. That's pretty cool. Well, if you're an organism that's single-celled and you're in a pond, maybe it'd be a good idea if you could see the light and you could swim to that place that was optimal for photosynthesis. Okay. So how do they see the light? They have an eyespot, and I've already pointed out to you, it's there on the slide. It's this orange thing that's asymmetrically placed in the cell. Okay, so the light shining on the eyespot, it stimulates phototaxis. So it stimulates this behavior of swimming towards the light. How does that work? How does that work? Well, I'm just going to blow you away here. These single cells, they get the light signal here. They get the light signal. It shines on the eyespot. Eyespot is on this side of the cell. There's a signal that goes from the eyespot to the flagella, kind of like a nerve impulse, and tells the flagella to change their behavior. So you saw that it was mostly symmetric, and they're doing the breaststroke in this direction. And when they are in the dark, they'll just go straight, all right? The light comes on. It's kind of like if you're in the swimming pool and you're going in this direction, and somebody you like is over on this side of the pool, and you want to swim over and chat with them, okay? So the light comes on. What do you do with your arms? What do you do with your arms so that you get to that side of the pool instead of going straight? Right? All right, so you pull harder with your arm that's away from your friend, and you kind of do wimpy strokes with the one that's on that side. And you turn your body, and then you swim straight towards them, right, with even strokes. All right. The most amazing thing is that Chlamydomonas, when the light is too strong, can completely switch the behavior and swim away from light. So why would light be too strong? You know, when you put your house plants right in the sliding glass door and the sun starts to move, like now in the winter, and it comes straight in on your plant is winter, and your plant dies because it's getting too much sun. Well, it's because it fries those green membranes that do photosynthesis. They just get so much energy from the sun, it just toasts them, turns them into brown stuff. So Chlamydomonas actually can sense when the light is too strong, and it swims the other way. Single cell can do this. Totally amazing. So how does this work? Well, remember this bit when you were looking at the cell that was doing the breaststroke way up close, and you saw that there was a little variation in the left one and the right one. What that variation does is as the cell is swimming straight, it actually twirls, and that little variation causes the cell to spiral as it goes forward. That's what this diagram is trying to show you over here. The cell is going to rotate in this direction. So as it swims, now the eye is rotated around here. It swims some more, and it's on the back side. And if you keep following it, it's going to come back around. So I told you that it beats its flagella 60 times a second, but it only rotates twice per second. So the rotation is a lot slower than the speed of the flagella. Okay, so this part's a little tricky, and we're going to go back here. But I'd really like you to understand this part, so we're going to do it slowly. If you have any questions, shout at me, wave your hands and shout, okay? In the next slide, we're going to be looking down at the top of the cell. And so you're going to be looking at the flagella sticking out sideways, and you're just going to be looking at the top half of the cell, and it's, you know, these are two-dimensional slides, so it's going to be a circle, all right? You're looking at the top of the cell, and this is the left-hand flagellum, and this is the right-hand flagellum. And what I'm telling you is if the signal could get from the eyespot to the flagella and tell it what to do instantaneously, then you'd want your eyespot to be right in line with your flagella, okay? You want it to be right there because you'd want this flagellum to start doing that big pull to turn towards the light, and this one to do the wimpy pull, right? But it takes time. It takes time for that signal to get from the eyespot up to the flagella and tell it what to do. How much time? It takes about 6/100ths of a second. That's the time it takes for that cell to rotate about an eighth of a revolution, and it's about 45 degrees. So if your light is down here and it hits the eye spot, it gets the signal, sends it to the flagella, but the flagella aren't going to respond until they're in this position, all right? And that's just where they need to be. If they're in this position after that time period, they're lined up just perfectly to turn towards the light. That doesn't quite, you don't quite get it the first time around. It's okay, we're going to talk about it some more. Okay. So the eye spot actually has to be, what we have to say that it has to be before the flagella, and that means in time, okay? It has to be in front of the flagella. It has to be seeing the light. It has to be in this position of the light before the flagella get there, okay? All right. So it has to be offset, what we call the flagellar plane. So this is what we call the flagellar plane, and the eye spot is actually positioned 45 degrees away from the flagellar plane. So this is really important. You're not going to get to the light if you don't have this offset for your eye spot. And the question is, how in the heck does the eye spot know where to be? There's only one in the cell. How does it know that it has to be at this place? Well, there's what I call a global roadmap. So, oops. Hint, think latitude and longitude. So we've got this globe-shaped cell, and I'm going to use these terms again about latitude and longitude. Okay, so I have to introduce a couple of other things before we get back to this global roadmap. And the one I want to talk about is called the skeleton of the cell. It's called the cytoskeleton. Cyto is for cell. The cytoskeleton. And there are different types of cytoskeleton, all in the same cell. I'm going to tell you about the microtubule cytoskeleton. This is a skin cell, could be from a human. And you have this network of these little tubules called microtubules. And they give the cell shape. It's kind of like the bones of the cell, right? Gives the cell shape. The other thing, very, very important, is these tubes are used as railroad tracks. They're used as railroad tracks to get things from the nucleus out to the edge of the cell. Really cool. There aren't things with wheels that go on the tracks. It's more like little molecules that walk, all right? There's the one that takes the photoreceptor to the eyespot. That's called kinesin. And the kinesin molecule looks like a human walking. It has 2 legs, and it walks along the microtubules like this. So it's really important, these microtubules, they not only give the cell shape, that sounds, interesting, but not that interesting, but being used as tracks, wow, that is really cool. This is a neuron. This is the cell body of the neuron. Remember I told you about the little dendrites? They're picking up the signals from other neurons. And then there's this long, long thing called the axon. And then it hits up on the next nerve cell on their dendrites. But see all these fibers here? Okay, these are microtubules. Fibers you see are called microtubules, obviously because they're inside of cells, they're not very big. They're little tubes, and the really cool thing is they're used as tracks, and it's really important in neurons because, these are really long cells. You got to get all that stuff out there that's doing the interaction with the next nerve cell. Your neurotransmitters have to be packaged and sent along with these tubular roads all the way out to the end. All right, so really cool tracks. They're called microtubules. If we blow it up to the molecular level, they are actually tubes. They have a hole in the middle. And they have structural strength and also are tracks. So these little kinesin molecules that I was talking about walk on the microtubules. They'll walk down the microtubules. Okay. Chlamydomonas has a microtubule cytoskeleton. And if we look, the protein, let me go back. The protein that makes up microtubules, there's 2 that are pretty much the same. They're very similar. These just look like blobs, but they make this really important structure. So basically 2 types of the same protein. So that's where the name comes from, tubulin, this protein that makes up the microtubules. If we use an antibody that has a blue fluorescent dye attached to it that recognizes tubulin, this is a tool we use, and all these pictures we're showing with fluorescence, the antibodies are directed to specific proteins. So this blue one is directed against tubulin, and you're seeing all the microtubules in the cell. So it looks like a fuzzy hairball, all right? There's some special ones, there's some special microtubules in here that have been altered just by one small molecule called an acetyl group. And it goes on a very specific amino acid in that tubulin protein. And it's a lysine, and it makes the tubules last longer in the cell, makes them really what we call stable. We actually have an antibody that recognizes acetylated tubulin and not all the other microtubules. And then we can see these really stable structures. And the structures I'm going to tell you about right now are the ones that are really important for this story. These are called rootlets. So there's these 2 rootlets have 4 microtubules in them. So they're bundled together kind of like a bundle of wheat, all right? But much neater than a bundle of wheat. And there's only 4 of them. So there's 4 in this one and 4 in this one. You can see that they're a little bit fatter than these, all right? They have 2 each. And then there's these funny structures in the middle called basal bodies. These structures are what the flagella grow out of. And they're made of tubulin also, all right? So this is a cartoon. I'm going to show you a lot of cartoons. These are the basal bodies that the flagella grow out of. And these are what we call the 4-membered microtubule rootlets and the 2-membered ones. This is the global roadmap, okay? This is the thing that's going to determine the longitude and the latitude of the cell. All right. More pictures and more designations, okay? So the picture I showed you before, it looks like the microtubule cytoskeleton was symmetric. We cut it in half, fold it over, and it would fold over exactly. But it's not true. Each of these structures has its own identity. I'll just tell you that all these green structures, and this is just a cartoon, are what we call the mother structures. So there's a mother flagellum and a mother 2-membered rootlet and a mother 4-membered rootlet. And the ones on this side, D for daughter, all right? No sons, no fathers, just mothers and daughters. Okay, this flagellum is the daughter flagellum. This is the 4-membered daughter rootlet and the 2-membered daughter rootlet. If we look down on the top of this cell, you'll see a lot of these maps. This is what I showed you before. We're looking at the top. We're looking at the top, and you see the 2 flagella, the 4-membered rootlets, and the 2-membered rootlets. And the most important thing is that the eyespot is always on the daughter 4-membered microtubule rootlets. So it knows that it's supposed to be there. How? How? You can see how we get really excited about this kind of stuff. I want to know how, even if you don't. All right. So mother, daughter, what does that mean? Why do we call them mother and daughter? When do cells make daughter rootlets, daughter flagella, and a new eyespot? Every time they divide, okay? So the daughter structures are made in every cell division. The eyespot goes away, and you're going to see a movie of this. The eyespot in the mother cell that's going to divide goes away. The cell divides, and it makes 2 new eyespots in the 2 new cells. You're going to see that with your very own eyes. Yes, we're going to see the video. Okay. So when the video starts, I want you to watch this cell, okay? And I'm going to try to put this down and see if I can stop it at critical places. We may watch it a couple of times. I think it only lasts about 17 seconds, so be patient. We'll start it. Okay, this is YouTube, so you have to get rid of all the advertisements. Haven't figured out how to do that ahead of time. I just want to point out, this is the cell. You can see a little wisp of a flagellum here. You can't see the other one in this picture, but here's the eye spot. And I want you to keep an eye on it. Keep an eye on the eye and see what happens to it. It's going away. Stop. Oops. Okay. So one of the things that I want to point out is the eye spot disappeared. You saw it disappear. And what we call the cleavage furrow, and this cell is actually going to end up being 8 cells When the cells are really well fed, they do multiple divisions all at the same time. It's a little annoying for me to show you, but just concentrate on these two, and you can see that what we call the cleavage plane or the cleavage furrow that's between the two cells goes right through the plane where the eyespot was. All right, now I want you to see where the new eyespots come. Okay, so we get 8 cells. And where are they coming? Yeah, on the outside. Here's one, here's one, here's one, here's one. Look here. Here's one, here's one. So these are brand new. They aren't anywhere near where the old one was. Everybody gets a new one. And they're on the D4 microtubule rootlet. All right, so we're gonna see that. Let me just see where we are in this talk. Wow, I am way over time, but maybe you'll humor me and we'll keep going. Is that okay? All right. Yeah, I'll get it here. All right. So this is just a picture from the side and from the top. Picture from the side, picture from the top. This is the side, this is the top. You're going to see a lot of pictures like this. This is what you just saw. Started out here, and then you saw the 2 eye spots in the new cell. This one disappears. So I'd just like to show you a little bit about how we figure things out. How do we figure things out? We get mutant strains that lead us to the gene, which leads us to the protein. The protein we can take and put into animals, usually rabbits. We get rabbit serum from the rabbits, and this is how it has the antibodies. And then we put a fluorescent tag on. It can be blue, it can be red, it can be green. And you'll see all 3. So we get the antibodies, and then we can see where these proteins are in the cell. So I'm going to skip this part. This is how we get the mutants. We mutagenize with UV light, and then we do a thing called enrichment, which we're just enriching for the cells that can't swim towards light, and we get the mutant cells. I've brought you my box, and I'm going to show you that afterwards. You can come down. This is my most important piece of scientific equipment. It's a cardboard box. I just want you to know that we've been using your federal dollars wisely. So I collected all my mutants with this box over the last 20 years. Okay, one of the things you saw in this video when we showed it before was that the dish on the right has a mutant strain in it that couldn't see the light. So this is just another way of showing it. We usually do it with tubes, and I'm going to show you in just a minute that we have a mutant in that box. The mutant strain that I'm going to tell you about is this multi-strain. The protein is required for the eye spot to be placed on the D4. If it's not there, it makes multiple eye spots. And they're in the wrong place, and the cells are all confused, and they don't know which side of the pool their friend is on. Okay, so this is what they look like. About half of the culture will have 2 eyes on the same side of the cell. Sometimes they have them on opposite sides. If you take an opposite side one and grow it up, there'll be 2 populations, some on the same side, some on the opposite side. They can't do phototaxis, and they have multiple eye spots. So it took us a long time to get the antibodies to this particular protein. Just to tell you that nothing's easy in science, it took us 3 tries. We kept trying because we knew it was a really cool protein. And this is what we saw. We labeled it with a pink fluorescent dye, And what you're looking at is the side of the cell. We're seeing the microtubule rootlets in green. And here's this multi-protein, and it's only on one rootlet. And we know it's the D4 because if we use a blue dye tag for the photoreceptors, the molecules that absorb light, here's the eyespot. It's right here. And it's at this junction between the end of the MALT1 track and the end of the D4. So this one protein, it dictates which microtubule rootlet it should be on. So that's the longitude story. And then it's this interface between where it stops and the rest of the rootlet that determines the latitude. This is the coolest thing I think I've ever worked on. Okay, so MALT1, that's the take home here. It dictates the longitude and latitude of the eye spot in this funny place that it has to be so it can do phototaxis, so it can be in the right place to do photosynthesis in the pond. I'm going to skip all of this and just tell you what we've learned. Eye spots form near the end of the D4 rootlet. They form at the end of that MALT1 track, and it's deposited on the D4. The part I didn't just show you, which is really complicated, it gets put on the D4 when so there's a daughter rootlet, right? It's a brand new rootlet in the new cell. As it's growing, this protein goes on the rootlet while it's growing. Totally cool. All right, so that's my story. And I just want to talk about this person is Telsa Mittelmeyer. She is really responsible for all of fluorescence microscopy. We were a lot younger in this picture. Full disclosure, Mary Rose Lamb started this project with me during my first sabbatical leave at the University of Colorado in the lab of Susan Dutcher. This is my graduate student, Joseph Boyd. Mark Thompson was my last graduate student. He just sent me a picture of he and his son going on Grant's first deer hunting episode with his grandfather in the snow. So there you go, there's another allusion to snow. All right, so that's the end. Thank you, and I'd be glad to answer questions.
Does anybody have questions for Dr. Dieckmann?
Is it a similar process that has a nose helping with a nose or an ear with an ear or that kind of thing?
So how you become a nose and how you become an ear is a little more complicated. So there's a whole series of things that happen. There's some big genes that called the homeobox genes that set up the patterns in our bodies. And the interesting thing is the homeobox genes are in the order on the chromosome, in the order that they're turned on to make your nose. So make the first part and then makes the next part and then the most elaborated part like the tip of your nose. So pretty cool. That's not what I work on, but it's, you know, development's cool. Maybe we'll get somebody here that talks about development. Yeah.
These cells you showed us, they have no nervous system or brain, but obviously it's all being done through the genome or through chromosomes?
Yeah. So the genome of Chlamydomonas is, it's not real small. It's, you know, it's about the same size as the Drosophila genome, maybe a little bit smaller. So all of the information for the proteins are encoded in the genome. They are single cells, and they don't have a brain, but the photoreceptor is what we call an ion channel, which is like the ion channels that work in nerve cells. So when the light is absorbed, it opens the channel, and calcium ions go into the cell, And that wave of calcium goes up to the flagella, and we don't know how this works yet to change their behavior. But there's some really cool motor proteins in the flagella called the dyneins, and they're responsible for this breaststroke shape. And their behavior has to be changed at that place. We just don't know how that works yet. So there are other really people that work on flagellar function that are really interested in how they change their shape. So I don't work on that. Yeah. Yes, they're only mutant in the one protein, and it doesn't affect their sexual behavior, and it doesn't affect their division, so we can keep them. I've had these mutants I've had that I told you about today were collected in the summer of 1991. So I've had them for a very long time. We get new ones all the time, but we've had these strains for a long time. And the cells are just fine. They just can't see. So it's like a blind person. They're alive. They just can't see. Yeah. Yes. Oh, that's a really good question. So the question, I'm not sure all of you heard, was what makes cells divide? So it's usually the same for all cells that are going to divide. You get to a certain size, and then there's a trigger for division. And it has to do with nutrition, a lot with nutrition. I showed you cells that divided 2 times, bing, bing, like that, and you got 8 cells from the original mother cell. If we starve them a little bit, not really starve them, but don't give them as much food, they'll only divide twice. You saw the other one only made 4 cells. And when we do these experiments, we only want to see 2 because it's too complicated to look at 4 and 8, right? To look at all the microtubules and stuff. So we don't give them quite as rich food. And then they get to a certain size and they only divide once. So we get 2 cells. And it's really true across all cells, all organisms. When they get to a certain size and they have enough nutrition, they divide. So it's not when they're unhappy, it's when they're happy. Yeah.
Any other questions? All right.
Well, I was fascinated by everything there. The fact that those critters can swim around in a crowded bowl like that and not run into each other. And then they can go so fast that I don't know how much energy they use per minute, but you'd think they'd run out of gas before too long as much as they move. And as small as they are, they must be at the bottom of the food chain. Who eats them, and what do these things eat?
Yeah, so that's a really good question. There are a lot of organisms that eat algae, many actually. There's some hypotheses about development of multicellularity. There are multicellular Chlamydomonas. So have anybody ever seen these beautiful globe-looking organisms that look like green globes? They kind of look like Christmas ornaments. They're called volvox. They're often on the front cover of biology, introductory biology textbook. They look hollow, they look pretty. Volvox are made up of like 2,000 Chlamydomonas cells that are in a globe. And there are other Chlamydomonas that only have 8 cells, and another one that has 16 cells. These are different species. And people are very interested in how they became multicellular. We're multicellular. So it's called the transition to multicellularity. And I think this is kind of where you're going with your question is, well, what I would like to tell you about is there's a hypothesis. I'm not sure I believe it, but that's how science works. And there are organisms that eat Chlamydomonas. And so the idea is if you're bigger, these bigger organisms that eat Chlamydomonas can't eat a Volvox. It's too big. They can't get it in their mouth. So they think that this might have been the evolutionary pressure to get these multicellular Chlamydomonas. So the organism that it's kind of like a little shrimp, they think. It's called Daphnia. It's like a little freshwater shrimp that eat algae. And I think something like it might have been pressurizing Chlamydomonas to become multicellular. Interesting hypothesis. I don't know if it's true. It's a really hard thing to test, right? It's so somebody's invention.
Questions for Dr. Dieckmann? All right. Well, thank you all for coming out tonight. Our next event is Thursday. Thursday, January 9th, with Professor Ryan Gutenkunst from the Molecular and Cellular Biology Department, and his talk will be titled I just had it written down. I'm sorry. It's genetic testing, hype, and reality. I know we had a few questions about 23andMe at the last event, and genetic testing questions before, so I think it'll be a really great talk. So January 9th at 6:30, and we hope to see you there. So thank you, and welcome me in applauding Dr. Dieckmann. Thank you.